Without further ado, we're going to launch into the GeneticMap keynote. I would be remiss, however, and I'm going to co-moderate the session, and I'm going to do so together with Claire Churchhouse. For those of you who don't know Claire, she's a fellow alumni from Oxford, where she has trained with Jonathan Marchini. She did a postdoc with David Altshuler in Boston, whereupon she met Ben, and then has become the scientific advisor in the Stanley Center. And since this summer, Claire is also working at the scientific liaison for ICDA. So Claire is going to co-chair this session with me. So first up on the podium to talk about science, from maps to mechanisms to medicine, is Ben Neale, who is on the EC in ICDA. Ben doesn't need a lot of introduction to this room, but for anyone who's listening virtually, Ben is a lead of ATGU. He co-leads the Psych Genetic Consortium. I'm going to forget all your titles, but there are many of them. And he's very well equipped to talk us through this first session. So thank you so much, Ben.
Thank you, Cecilia, for the lovely introduction. And thank you all for joining us today. It's truly a pleasure. This is a meeting that's delayed by, I guess, 2 years and 9 months. I think it was April 2020. We were expecting to have this meeting, and I can remember sitting in Eric Lander's office saying, we're going to have to cancel. And he's like, what do you mean? And I was like, it's not going to get any better, and it's going to get a lot worse. And little did I know how true those words would be. So let's talk about maps to mechanisms to medicines. We've been at this kind of genetics effort for quite a long time. And before I dive into the science, I just want to orient us to our principles. I think when we come together to try and tackle really big, complicated problems like why do people get diabetes and how do we make it better, or what is actually the biological basis of schizophrenia, these are forbidding challenges. And so there's no way that one person, one group, one lab is going to magically solve these difficult problems before us. Indeed, it will be a community-wide activity that makes the durable progress in understanding how these diseases arise and how we might intervene to help people move beyond these particular conditions that we're interested in studying, particularly the common complex diseases. And so when working in a community, I think it's always important to talk about what your values are and what your principles are and commit to those things as a way to proceed, because there are lots of structural inequities in our society. There are many problems before us, but there is that tremendous hope in science that we might discover something new about the world, something that might help us realize a new way of approaching a problem. And so with that in mind, we put our heads together and developed a set of principles to really organize our behavior in working together. And I think it is just an important thing to take a moment and reflect on them. I'm not going to read the slide. I'm sure most of you have read through it. And if you're not so interested in the value side of things, just wait another 10 seconds, and then I'll dive into the scientific content of the meeting. But yes, ICDA is about the community. This room, everyone online, everyone around the world that is committed to this mission of taking genetic information, using it to learn about disease with a hope of improving the quality of treatment, management, diagnosis, outcomes, course, all disease-relevant considerations. That is what we believe is possible. So the core scientific idea is one that starts with people. It starts with, diseases like schizophrenia or diabetes, and, goes all the way down to the genetic variant level, this lovely sort of schematic of going down to the variants, collecting the genetic information, linking that to phenotype to do that GWAS that we've gotten really good at, so good that it's become kind of turnkey and almost rote at this point in time. That GWAS has taught us that human diseases are very, very complicated things. There are lots and lots of biological parts to this story, and we are not in a position to understand or interpret what those genetic clues are trying to teach us. But there is the possibility of taking the emerging technologies in the biomedical research landscape to drive beyond just the association of genetic variation to individual diseases, thinking about learning about what the genes are, what the cells are, what the processes and physiological functions are that go to make up these disease entities in the population, to find those mechanisms to hopefully think about rational basis for novel therapeutic intervention. You see this little star, and there's like the Clausnitzer little reference there. So this is a figure that Melina, I think, made with her husband a long time ago, on the back of the kind of, one of the first, I think, nice V2F studies in the literature working on the FTO locus in diabetes. And indeed, in ICDA, we have efforts focused on genetic maps, trying to bring together the world's data to really power the discovery of human health and disease phenotypes, but also a series of projects that are not quite in the we have a logo yet stage. So I just wrote down their names on the right-hand side of the slide, traversing the body map, cell map, blood single-cell RNA map, and a disease map. I'm just going to try and set the stage for what we're going to discuss over the next 2 days. And this is very much intended to be a dialogue. We want to have an engaged conversation about where we are, where we're going, what experimental paradigms we think are going to actually move the needle on this question of going from maps to mechanisms to medicines. Okay, so let's start with some genetic maps, and I'll dive into a little bit about the science. So the Global Biobank Meta-Analysis Initiative was an initiative that largely spawned out of ICDA during the kind of convening functions in one of the various meetings. I think it was the meeting in Helsinki. We really got our kind of heads together about what was possible. Now, obviously, large-scale population cohort biobanks, things like the UK Biobank or All of Us or many of the other biobanks that you see on this slide, have sprung up across the world. And they're not, fully representative of the world's population just yet, but they do represent a huge amount of genetic information and opportunity for trying to work together to power the investigation into potentially any human disease. I think as long as we can collect high-quality phenotype information, put it together with high-quality genetic data, we can start to make progress either through, say, genotyping to map the common variant piece, or maybe I'll talk about sequencing in a moment. And that's exactly what we did in GBMI, that which we launched with Mark Daly and Kristen Willer, and now Ida Soreka is taking over for Kristen as she's moved to Regeneron from Michigan, all spearheaded by the very, very talented postdoc Wei Zhu, who's been a true pleasure to work with. And the kind of first wave of GBMI papers was published in Cell Genomics just a couple of months ago. And here's one GWAS, one example, one kind of indication of what may be possible if we try and really bring these world's data sets together. And so here's a picture of the genetic discoveries for asthma. You can see the kind of blue line is the set of previously reported associations from the international collaborative efforts in asthma genetics that already spun up. And what you see below is the red line, and you see this previously or the kind of potentially novel associations that we had not yet mapped because we didn't have sufficient statistical power to really uncover those associations. This was work led by Christian Swo, a PhD student, working with Alicia Martin and Mark Daly and many others, to try and see what we can learn about that kind of disease association phenotype. Now, even with GBMI and even with the expansion in sample size, or even thinking about something like diabetes, where there's much larger samples and a much greater investment already committed to understanding the genetic contribution to that particular illness, we are far from saturating common variant discovery. Height is maybe just about there, although that's just maybe found at the loci rather than understood what the fine mapping common variants are specifically. So it's not even that that's job done. It's more like we know the regions of the genome, but we don't know the variants at this point in time. But for disease phenotypes, for the things that are the public health challenges before us, the things that cause so much suffering around the world, there we still have a long way to go. And so, working together is a core component of that. Now, I did mention that fine mapping and trying to understand what the common genetic variant or variants are at a locus that is driving the genetic signal that we find is still very hard, / I would even say unreliable at this point. So this is some work from Hilary Finucane and Ran Cui, looking at the replication failure rate. So the idea behind the replication failure rate is that it's hard to have ground truth for something like fine mapping. We don't actually know what the local genetic architecture should look like for common variant associations at this point in time. We don't actually know what that ground truth is, because frankly, it's not really been worked out for terribly many loci, and maybe even amongst a handful of loci where there is some effort to do that. I think there's still a lot of debate in the community and in the literature about the kind of reproducibility and accuracy of that. And so Hillary had this really magnificent idea of taking UK Biobank, downsampling it to 100,000 individuals, running a GWAS, identifying genome-wide significant loci, and then estimating what the posterior inclusion probability is using the current state-of-the-art in fine mapping algorithms to try and resolve statistically where that genetic association is driven. She then scaled up the data set to 366,000 individuals, the broadly white British and genetic ancestry group, broadly speaking, for reasons of LD and whatnot. But just this is like trying to do that question of what is that common variant, and ask the question of how well calibrated are the posterior inclusion probabilities from that 100K analysis, right? So if fine mapping is doing its job properly, then the things that we're making a bet on that are the driving SNPs for the association should hold up when we increase the sample size. That's good statistical theory in practice. And here's what we see. So taking the posterior inclusion probability SNPs that have a 90% chance of being a real causal variant, you have an expectation that they should not replicate a little bit less than 10% of the time, because there's obviously a distribution there. You take the posterior inclusion probability that's coming from the actual algorithm. And empirically, that's nothing could be further from the truth, right? You see replication failure rates up in the 15, maybe 20%, depending on the algorithm, depending on the threshold, depending on what you're looking at. And that just underscores that even though this is like best case scenario for a trait mapping exercise, right? So this is a single cohort collected in the same way, genotyped on the same platform, imputed in the same technology, with the same principal components included to control for ancestry, in a large sample size so that the genetic structure is reasonably well understood, right? This is as good as it gets right now. And even in this circumstance, we are seeing pretty dismal replication failure rates. I don't think that this is going to be any better for the meta-analyses from the GWAS for things like schizophrenia or diabetes that are drawing together cohorts from across the world. Indeed, I would fully expect those to be worse because of the uneven data quality questions and the differences in the quality of the control of ancestry and all kinds of technical issues that actually make this probably rosier than it would be if we're trying to apply these algorithms in the context of, say, the meta-analysis for type 2 diabetes. So Hillary, not satisfied to leave it there, then generated a potential solution, include another term, a little infinitesimal component in the fine mapping model, and it gets a little bit better, right? So if you basically smear out the association signal a little bit and say that there's going to be some kind of uncertainty everywhere at some level, that does slightly improve the replication failure rate, maybe probably because it changes the SNPs that are on the bubble that are a bit uncertain. But even there, we're not in a place where this is a replicable, reproducible, kind of honest, reliable way of identifying what the causal genetic variants really are. Now, maybe not all is lost, although there are some lessons on both the good and the bad side here. So you can take a look at this replication rate across the 100K versus the 366K. And then you can stratify the variants that are being included in the fine mapping solution as a function of the functional annotation. And this is just a very broad brushstroke annotation. We have coding variants, so really just the missense mutations and the putative loss of function mutations. And then putatively functional variants, so things that are in enhancers or synonymous mutations or in the untranslated regions, those sort of putatively regulatory variants, not that we understand how to interpret those regulatory variants at this point in time. And if you look at the things that are replicating, you can see that there's pretty strong enrichment, particularly for the coding variants, although it's worth recognizing that the non-replicating posterior inclusion probability SNPs are also slightly overrepresented in coding variants compared to the background variation that you see genome-wide. And that's because functional variation hangs out in genes, and LD is pervasive, and it's really hard to get these algorithms to be really, really accurate. So functional information, being able to functionally interpret what genetic variation is, should help us interpret these kinds of genetic associations. And the lack of a clear understanding of what genetic variation is doing in the non-coding region of the genome in particular, I think is certainly impairing our ability to identify what the causal variant is and use that as the starting point for these sorts of mechanistic explorations. We also recognize that there are cohorts all across the world that are not only being genotyped, but being sequenced. And so in collaboration with Cecilia, Konrad Karczewski, and Duncan Palmer have put together this BRAVA effort, or the Biobank Rare Variant Analysis Network. I like that name. I felt pretty proud of that name. I thought it was a good one because we got a nice little emoji logo to boot and everything. But again, bringing the world's data together in a shared mission of trying to understand that genetic basis in as systematic and consistent and interoperable way as possible, that is a core value of our community as human geneticists, and I'm delighted to say that we're continuing in that core value and continuing to try and make progress in understanding that fundamental genetic basis of many, many, many different illnesses and diseases as they come. But if you just sort of reflect for a moment on BRAVA, you reflect for a moment on GBMI, there's some pretty notable gaps. Not a lot of biobanks in Africa. There are some. It's starting, and H3 Africa is putting things together to try and bring that forward. But there's really no major biobanks in these efforts at this point in time in Central and South America. And so as part of that, you're going to hear today, I think, yes, about AdMixMap that Andrés, Moreno, Estrada, Ricardo Verrigo, Isquia Sánchez Lopez have been working on in terms of trying to realize opportunities across this part of the world to increase the underlying exploration of genetic variation. Because remember, each of our different genetic ancestries has a slightly different set of genetic variants that are slightly more common in that group of individuals, which gives us a new opportunity to power a genetic association test and learn something new about new genetic variation. And so, there's a good scientific motivation for that, but there's also a, I think, ethical and social justice imperative to really address the longstanding inequities, and so efforts to bring us forward into this kind of space where everyone can participate in the science and they can be true partners in our journey together. That's also a goal that we share and reflective, I think, of the principles that I started the talk with. So that's kind of where we are on discovery and where we are in trying to understand the genetic basis of human diseases. But that's not the only question before us, right? We also have the fundamental question of how do we go from genetic variants to the effector transcript? What gene matters? How do we figure that out? And so you'll also hear about BodyMap during this meeting, which is an effort to try and close that gap between genetic variants to genes. So how do we actually make that leap? So Tuli, Kristen, and Andrés have been working on a proposal to do a multimodal investigation, multi-tissue in the same individuals. I think there are a lot of open questions about the portability of insights that you can make from one cell or tissue population across cell and tissue populations. As someone that works on psychiatric disease, I'm pretty dismal on the opportunity to get a huge amount of live brain tissue. Just doesn't seem like it's going to be on our roadmap anytime soon to get a very large sample of that. So I desperately want to know how transferable insights from different cellular populations are into being able to interpret what the consequences of common genetic variation really look like. So you can see on the slide some of the technical details and things that are specced out in terms of the scope of this particular proposal. But again, it's trying to move us beyond just that genetic variant and increase our ability to determine what the consequent gene is that matters. Genes are part of the story. There's another way to maybe jumpstart this kind of investigation that also relates potentially to some maybe blood-related disease traits or entities. So Nicole Soranzo, along with other colleagues at the Sanger and actually other institutions in the UK, has worked on a sort of initial pilot from UK Biobank to do single-cell RNA-seq from blood, from whole blood, do the cell sorting, do the single cell profiling, and then you can start to do the genetic mapping there. Obviously, UK Biobank brings tremendous added value to this kind of enterprise insofar as we know an awful lot about these people's medical histories, about what diseases they've had. There's a huge amount of assessment information that helps understand the generation of these mainly late onset diseases. I think that's the kind of primary focus for UK Biobank. And so Nicole will talk us through what is possible in a kind of blood cell map way that maybe doesn't address the question across tissues, but does give us the opportunity of going very deep in a single tissue to see how much we can really learn from this kind of paradigm. It's good to go broad. It's also good to go deep. We try and do that across many of the different projects and pitches that you're going to be hearing about over the 2 days here. So that's linking variation to genes. But we don't just want to stop there, right? Like, there are other important questions before us. How do we go from these variants to cellular functions? How do we understand the actual disease processes as they are generating? How do we understand what's going on in a cell explicitly? And so Gosha and Molina and Blagoya have been working on this cell, maybe function map is what I call it, or functional cell map, something in that kind of space, where the idea here is that you again have genetic diversity in lines. So it's not just about doing a demonstration proof of principle experiment in an in vitro system. It's about taking maybe 100, maybe 200, maybe 500, maybe scaling up to 1,000, and taking, induced pluripotent stem cells or other progenitor cells that are readily kind of accessible, so things like adipose-derived mesenchymal stem cells, coaxing those cells down their developmental path into a more mature cellular state, and then subjecting them to a series of assays in order to fox out exactly functional readouts from these cellular populations. And so that might be things like transcriptomics, so you can look at expression. It's nice, unbiased, generic, very easy to do, relatively speaking, these days. But expression is like not function. Right? It's maybe related to function. You can maybe use it to interpret something about cellular function, but it's not telling you about, ion flow through a calcium channel or about, T-cell activation. Like, maybe you can get the signature of it, but you want maybe some other features about the cell to try and interpret and understand what that cell is doing. And so maybe we want to add some labeling of surface proteins and do some imaging in that kind of way. Or morphological and cellular phenotypes, where you can do a stain, an image-based capture, and then use that to create quantitative readouts of actual cell functions. Let's GWAS those. Let's use genetic regulation of these traits to convince ourselves that what we're observing is not just technical variability in the dish, but actually biological variability. Genetic regulation is an opportunity not only to demonstrate that this is actually recapitulating true biological variability, but it is also an opportunity to take that genetic regulation, use approaches like Mendelian randomization to link the cellular functional readout to the disease endpoint. And then you can say, hey, look, I now have this, maybe adipocyte-like cell population. Maybe I can figure out what the blood glucose or like the glucose gradient response is to for that adipocyte population. Maybe I can do the genetic mapping of that. Maybe I can relate that to certain phenotypes around obesity or consequences of obesity or other fundamental aspects of disease biology. And you go look up in the GWAS of the disease in question to see whether or not the genetic regulation is shared. And in the event that it is, that's now actually a very good, solid bet that this cellular model is recapitulating something about the disease process in question, which then makes it perfect for screening. So you can kind of see how this might actually start to get us from genetic mapping to cellular mechanism to now being the kind of base source material for medical development. But the thing about Cell Mapper, which I love as an idea, I'm really excited about that, man. I was just talking with Gosha and Molina and Blagoya forever about that. It's endlessly entertaining. The thing about it is that diseases are not generic, right? Sequencing is generic. Transcriptomics is generic. Diseases are unique. They are all different. We have classified them and separated them and sorted them into different categories, and maybe some of those lines aren't quite right. We think about that a lot in psychiatry, as you imagine. But because of the kind of very bespoke nature of disease, it seems also very important not to lose sight of what our end goal is. Our goal here is not just to do fancy science. That's not really our express goal. Our express goal is to do science in service of the mission of understanding human diseases. And so John Ryu has been working with a kind of laundry list of collaborators across 3 different disease areas to try and articulate what those kinds of bespoke follow-up experiments that you might want to do to interrogate specific disease entities. So perhaps you want to take a look at some solid tissue profiling from individuals that have specific disease. So an inflammatory bowel disease, for example. You might want to look at the cellular epigenetic landscape in inflamed tissue versus non-inflamed tissue in patients. That's not something that we're going to get out of BodyMap because IBD is a pretty rare phenotype, like 0.1%. So it's not something that you're going to just get along for the ride. You need to be purposeful and thoughtful and direct with respect to the disease areas. And so that's the idea behind DiseaseMap, is to try and develop sister scientific projects that can capitalize on the foundational resources that I've kind of tried to walk you through in the beginning of this talk to accelerate our understanding and interpretation of these pioneering disease areas so that we can ensure that these resources that we're building are actually reflective and responsive to the needs of the communities trying to interrogate and understand those specific disease areas. And so with that, I will say thank you, and I guess open the floor for a question or two. Is that about right? Excellent.
Perfect. Thank you, Ben. So I think you set the stage for the next 2 days really nicely. I'm curious if there are any questions here. I want to flag to all the remote attendees that you can submit questions via Slido. You go to slido.com and you enter a code, which is 5066. I feel a bit like a bingo readout person here with this microphone. So 5066 is the code. And if anybody want to GoFundMe the project, a $1 pledge would... Sorry, we have an arm up over there. Please, a question? No? You just stretched a bit. Mike, no, yeah.
Ben, you laid out very nicely the vision for all these maps, and I was intrigued by the fact that, let's say you generate a single-cell data set from a case-control cohort. You could actually achieve the goals to some extent of the gene map, the cell map, the disease map, or body map, and so on. How distinct are these efforts, really?
It's a great question. So, just as a reminder, ICDA is not doing projects. We're just trying to convene the community to come together and rally around shared ideas. And so there's tremendous appetite for us to also try and partner with efforts that are going on in that kind of targeted way to bring that data on board into the broader landscape. If you think about, how useful the 1,000 genomes expression data sets were, in the early days, they were pretty transformatively useful in a number of offspring kinds of studies. And so we perceive the value of building these resources for trying to get our hands around exactly what we need to learn, how much we're actually going to learn about the genetic regulation of these things. But we're extremely open to opportunities to collaborate, share the data from these efforts, to power those investigations in the disease-specific ways as well. So keep it coming, I guess, would be my general response to that line of questioning.
I would like just to add that in the GeneticMap efforts, there are individual biobanks that have, genotyped and/or sequenced their cohorts. We're coming together as a grassroots initiative and sort of doing analyses across stakeholder groups. In BodyMap, there would be a global sample collection, which is something that isn't available today, and it's something that is quite tricky from an ethical, moral, legal, and logistical perspective. So that's a non-small feat to do. In CellMap, as you're going to hear from Milena Blagoje and Gosha. It's an IPSC initiative, so it's quite distinct of that, but BodyMap will be a foundational resource, we hope, that will serve the community. So the IPSC cell work is quite distinct. And then that of DiseaseMap, it's an umbrella of projects in various diseases, because various diseases have reached differently far in the sort of maps to mechanisms to medicine sort of endeavor. And I am certain that DiseaseMap will draw upon and collaborate with BodyMap and CellMap, but it's quite distinct, as Ben said, in the type of cells and samples that you need to collect. So I will say that they're intertwined, but they are distinct and sort of codependent, but not the same.
Makes sense. Thank you.
All right.
Do we have any questions from the Slido? And the code is 5066.
We do have one question that's come in from Slido. So can ICDA help in any way to establish biobanks in parts of the world where they do not already exist?
That's a great question. I mean, I think the sort of trying to coalesce the community and the efforts that the Latin Genomes Group have done and what ADMIX is doing speaks to the enthusiasm for helping to facilitate and address these inequities and gaps in where biobanks exist. So I would say certainly, and if you're excited to do so, come and participate, reach out. We're happy to help try and figure out how to be most helpful to realize the dreams and ambitions that you have for doing so.
We also have a biobank sort of advisory group chaired by Rory Collins, and he is intimately engaged both in H3 Africa and in Latin genomes and so forth. And then, of course, Andrés and Ricardo and other constituencies in ICDA can maybe talk a little bit to your experience. So what would you say about how ICDA can help, Andrés?
Sure, and definitely we will hear more about that in literally a few minutes. I would prefer maybe to wait for the moment to speak in detail about that. But definitely many opportunities are coming up to contribute to these global efforts and increase representation.
Yeah.
Wonderful.
So if no more questions, we're going to thank Ben again.
With that, I'd like to introduce our next speaker, Dr. Slavé Petrovski. Dr. Petrovski is the Vice President and the Global Head of AstraZeneca's Center for Genomics Research. He's also an honorary professor at the University of Melbourne. At AstraZeneca, Dr. Petrovski leads a diverse team of global talent that, in partnership with AstraZeneca's therapeutic areas, informs various stages of drug discovery to development through the use of human genetics.
Excellent, pleasure, and thank you for the invitation. It's always a challenge to speak after Ben. He set the benchmark so high now, so I'll try to keep everybody as engaged as Ben did. My 20 or 30 minutes presentation is really going to be about giving a little bit of a flavor of what the industry's perspective of human genetics is. How do we leverage the vast volumes of human genetics being generated across the globe to impact various stages of our drug discovery to development pipelines? And I think one critical point I want to mention right now rather than later is that effectively all the data that I'll be presenting in my slides aligns to the principles that we just heard from Ben and Cecilia. These are multiple pharma peers coming together with common goals, common visions to generate data that then is also available to the global research community to benefit all medical R&D across the globe. So, I come from AstraZeneca, as was just mentioned. I'm an employee and stakeholder. And at AstraZeneca, we have a diverse portfolio of disease interests spanning across primary, secondary care, and recently with the acquisition of Alexion, even in rare diseases. Back in 2016, AstraZeneca made a big strategic announcement in terms of its Human Genetics Genomics Initiative. This is a 10-year initiative with the bold aspiration of analyzing the sequences of up to 2 million individuals, including participants from our very own clinical trials, which gives us that really invaluable interventional component. We look to integrate that foundational layer of genomics, again aligned to some of the ICDA principles, with other modes of dynamic omics on those same individuals, where we also have linked health records and outcomes data. At AstraZeneca specifically, we have been focusing building scalable, robust informatics and analytics platforms, and we partner with various external parties to generate the data, either in collaboration or as kind of more of a fee-for-service platform. And we focused on next-generation sequencing in our strategy because in addition to obviously being able to capture the common variation that we capture well with arrays, it also captures the rare and ultra-rare genetic variation that's been less well studied in terms of its impact on common complex diseases, at least until recently. And just, final point, to be able to deliver this bold ambition, we recognized, again, aligned to ICDA strategy principles, this isn't something that we could achieve in isolation. And so we have an extended network of academic and global biobank and industry partnerships to help us achieve our goals. And effectively, what we want to achieve with the human genetics data is probably not too dissimilar to across the globe in terms of R&D. We want to better understand the underlying biology and mechanisms of disease, especially for those diseases with large unmet clinical needs. We want to use human genetics to help us identify the new medicines of tomorrow. We also want to use human genetics to support selection of patients for clinical trials and precision medicine strategies, effectively matching patients with the medicines that are most likely to benefit their underlying mechanistic cause of disease, and also inform life cycle and indication opportunities for our medicines, really capitalizing and maximizing the potential patient benefit for the medicines that we are developing. And so now I've got a really basic slide, and I apologize in advance, but it does set some of the foundation for the next few slides, so I wanted to cover it in terms of what do we already know or what do we kind of intuitively recognize. So we recognize that rare variants play an immensely important role in rare diseases. We know this through Mendelian studies, through familial studies, and so forth. We also acknowledge that most common complex traits have been well studied and continue to be well studied in terms of understanding the contribution of common variants through genotyping array technologies and imputation. And we also recognize that when we identify variants which have a large effect, large odds ratio, large clinical effect size, they're overwhelmingly enriched for variants that are lower frequencies in the general population. What we haven't been able to systematically assess, at least until recently, has been what is the contribution of rare genetic variation in the context of common complex traits. And with resources like the UK Biobank and other global biobanks that are coming available, sequencing-based data sets, we're now starting to be able to answer some of those questions. And I'm going to go through a few slides of our experiences using the UK Biobank exome sequence data. So this was an article that was published last year at the time on 300,000 exomes from the UK Biobank. Our public portal, which houses all the summary stats from variant, gene, and phenotypes, also now is updated with the 470,000 exomes release. But the data hasn't really changed that much in terms of some of the summary insights. So in terms of studying individual variants at a variant level and focusing on the coding sequence from the exomes, these were some of the observations that we had. So on the left panel, again, probably intuitively very familiar and as expected, but actually before I go into this, one of the important aspects here is that this is a reflection on a summation of tens of thousands of phenotypes. So it really gives a kind of generalizable understanding of what are the effects at the individual variant level across a broad spectrum of disease rather than any one individual disease. And so if we look at the far left side, effectively what we're seeing is, and I'll focus on the dominant genetic model there, about 25 to 30% of the statistically unequivocal genetic signals that were identified in the sequence data with the linked health records were not identified in the array and microarray data. And imputed array. In the middle panel, again, intuitive and what we already recognize, but again, nice to see, when we dichotomize the statistically significant variance based on common or rare here, just using a threshold of 0.1% math, what we see is that for the common variants, they're heavily enriched for variants that are significant but have very modest effect sizes, whereas for rarer variants, we see that greater distribution of increasing effect sizes across that spectrum. And in the right panel is what we're seeing in terms of novelty at this particular snapshot. And for variants that are annotated as protein truncating, where we achieve significance, 88% of them were already identified in one of four existing catalogs, so ClinVar, GWAS catalog, I think ThinGen, and I forget what the fourth one was. And then about 12% of the protein truncating variants hadn't been previously ascribed pathogenic to a particular disease. And for missense variants, that kind of novelty aspect increased to close to 30%. And this is probably also not too surprising given our greater confidence in ascribing pathogenicity to variants that have a protein truncating effect than variants that have a missense effect, given the wider spectrum of potential biological effects of missense variants. But even at 500,000 individuals and with sequence data, there are some variants that are considerably rare and some variants that are private to that pedigree that are not well captured statistically at an individual variant level. And so there are many frameworks out there that try to capitalize on all those ultra-rare variants and understand whether, as a class of variants in a given genetic unit, for example, a protein coding gene, there is an enrichment of, or a depletion of that class of genetic variation in cases and controls. And we refer to this framework as a collapsing analysis, but there's also gene burden and various other flavors of it, but they're all effectively trying to do a similar thing, and that is to assess the aggregate contribution of rare genetic variants in a genetic unit and determine its consequences or correlations with disease outcomes. And this is just a table taken from the Nature paper. We've got about a dozen models that we adopt, and this is in no way prescriptive. There are many ways that one could adapt these and many flavors. This is just a reflection of what we, through our experiences, have found have been quite impactful in terms of adding value to the kinds of analysis that we do. And I'm not going to go through all these. Maybe I'll just highlight the 5 columns in terms of the key references, kind of filters that we adopt. So we like to use an external allele frequency filter for our upper allele frequency, and we found GNOMAD to be quite useful for that. But of course, we also use an internal test cohort allele frequency upper limit as well. So that combines cases and controls and has a upper limit of minor allele frequency for the various rare variant collapsing models. Variant effects is obviously important and quite powerful within protein coding sequence. You have to be a bit more creative when you go step outside of the protein coding sequence. And then we have 2 additional filters focusing on the missense variants. One is Revel, and there are many others. And effectively, it's a score that tries to predict the deleteriousness of an individual missense variant. And then there's missense tolerance ratio, which is a score that we constructed, which helps us identify subregions of genes that seem to be in the general population preferentially depleted of missense variants, and then you can make that assumption or hypothesis that maybe identifying missense variants in these potentially critical regions of the genes might have greater clinical impact and let the data speak for itself. And when we adopt these across the UK Biobank phenome, if you like, this is some of the summary stats we get from a gene-level collapsing analyses. So on the x-axis, we've got the various different ICD-10 chapters. We anchor our phenotypes to these because it's quite convenient, given the ICD-10 chapters are well catalogued in the UK Biobank. Each dot represents a gene-to-disease relationship, so not a variant. It's an aggregated effect of various types of rare variants in that gene correlating with disease. And each dot above that dashed horizontal line is a statistically unequivocal link between a human gene and a human disease. And you see a nice spread across the various disease classes, which was really encouraging. But also, when we look at those genes that are above that dashed horizontal line, what we found is that they're about 7.4-fold enriched for genes that are known drug targets for medicines that are FDA approved, really exemplifying, I don't need to preach to this audience, but really exemplifying the power of human genetics for target selection in drug discovery and development. But then, there's some other questions that come up. Okay, that's great, we've done these gene-based analyses, rare variants, but really what are we capturing that we didn't otherwise get from just looking at individual variants? And for the binary outcomes, in our experiment, if you like, so these are the case control analyses, actually 82% of the signals that we discovered in terms of gene-disease relationships were not recapitulated by an individual variant in that gene correlating with that disease or a similar disease. So it's adding a lot of additional value beyond what we get from studying individual variant effects. That proportion drops quite dramatically down to 40% in quantitative traits, and again, probably not too surprising to many in the audience. We're much better statistically powered with quantitative traits, so the individual variants are able to pull out that significance, and you don't need the aggregate effect of individual variants. And one of the, I think, quite enlightening summary stats that came out is if we focus on the binary traits, these case control analyses, And again, as Ben mentioned, UK Biobank is heavily enriched for common complex diseases, late onset. What we're seeing is that the median effect size odds ratio is about 12.4. So that's quite a respectable effect size for the contribution of perturbations in these genes having on increased risk of disease. And then finally, again, aligned to some of the principles, as we recognize, the UK Biobank is about 90 to 95% European genetic ancestry. But when we included this additional, even incremental 5% from the non-European genetic ancestry, we identified multiple dozens of gene-disease relationships that didn't achieve significance when we focused just on the European data. So again, just that 5% increment, but from diverse genetic populations, really boosted the signal for a few dozen of these relationships. And now I'm just going to zoom in on one. So this is type 2 diabetes. It's a very, as we all recognize, common complex trait. In the UK Biobank, at the time of this particular snapshot, there were 32,000 cases and about 260,000 controls. I'm just going to focus on the rare protein truncating variant model. And what we identify is two statistically unequivocal signals and then quite a number in the suggestive range. And for illustrative purposes, I'm going to label here all the genes that achieve the p-value less than 10 to the negative 5. And when you look across these 8 genes, you see actually even those genes in the suggestive range, so they're not yet sufficiently powered to achieve significance, they're strongly enriched for known positive controls. You've got your HNF1 alpha, HNF4 alpha, TNRC6B, etc. Really, again, encouraging in terms of the robustness and the cleanness of this kind of rare variant collapsing framework to pick up real signals and have highly ranked, top-ranked signals that are positive controls. And then there were some others that we as a consortium and community unraveled through the exome sequences that weren't evident in previous array-based studies. And IgGIF1 is one example, and that's driven by rare protein truncating variants spread across that gene, and collectively they have a odds ratio about 4-fold in terms of increased risk for developing type 2 diabetes. But I want to focus on another one, which is that last one on the far right-hand side, the X chromosome here on MAP3K15, because even though it's absolutely precious to identify novel genes that contribute to increasing disease, and help us understand what are the pathways and the underlying biology that we could then look to try and mimic or try identify therapeutic intervention opportunities, it's undoubtedly much more accessible when you identify a human genetic target that appears to be protecting from disease. You've got this large in vivo experiment where natural inhibition effectively looks like it could be protecting individuals from developing these clinical diagnoses. And so there are many examples of this across literature, and I've only highlighted a handful here, and I apologize to any that are missing in here. It's just for illustrative purposes, but the path from these human genetic discoveries of natural inhibition protecting human population from developing these diseases to developing medicines that then benefit patients is a well-trodden path. And MAP3K15 looks like it's very early, but looks like could potentially be, in the future, one of these potential opportunities. We see that individuals that have a natural loss of function variant in this gene have about a 40% reduced risk of developing clinical type 2 diabetes. And when we look across the various data points, so this is based on the 400,000 UK Biobank exomes, we see that the signal first emerged from a recessive genetic model because it's an X-linked gene, and males who have genetic perturbations in this gene are effectively homozygous, and so the recessive model in this context tends to be a bit more powered than the others. And when we looked at the recessive PTVs plus predicted deleterious missense variants, we saw a signal for clinical diabetes and glucose reduction, again aligned with the argument that it could be protective of clinical diabetes. And then when we focused just on the PTVs, we lost some of the statistical power there because it was about 20% of the qualifying variants that were 20% of the observations. But the effect sizes improved in the direction that one would expect when you remove the missense and you just focus on protein truncating loss of function variants. And then we looked at 2 other resources that we had available at the time. So one is the Mexico City prospective study. It's about 130,000 exomes, and we're able to see similar relationship there in that population, and also in FinGen. And I think most of you are familiar with FinGen. This isn't a sequence-based study. It's a well-imputed genotype-based study, but the beauty of the Finnish population and various other populations that are bottlenecked is that it allows rare variants to become more common, or you identify generally rare variants in European populations and other populations more at higher frequencies in these bottleneck populations. And one of these protein truncating variants, this stop gain variant, was about 2 to 3-fold more common in the Finnish population. And when we looked at that particular protein truncating variant, it again effectively replicated what we saw in the UK Biobank and Mexico City. So collectively, again, statistically unequivocal when you look across the 3 biobanks. But then that starts to ask other questions. Well, okay, how does this fit into what we already know about type 2 diabetes and the genetics of type 2 diabetes? And one obvious question is, what is the impact of BMI? And what was quite interesting and intriguing about MAP3K15 is that the protective effect appears to be independent of an individual's BMI. And that was perhaps one of the first early clues that maybe the underlying mechanism is independent of the typical insulin resistance pathway. Also, because it's on the X chromosome, you've got a lot more homozygous carriers, and what we see is actually heterozygous female carriers have about half the reduction in glucose levels as what we observe in the male homozygous carriers, so potentially an additive effect there. And one of the other beauties of these big biobanks, as I'm sure many of you recognize, is that in addition to the phenotype that you're interested in, you also have access to a breadth of other phenotypes. And so you can start to question and probe these genetic perturbations. Are they having other impacts on other potential clinical manifestations? Are there any potential safety liabilities? And it was encouraging from the human genetics data, at least, that in the MAP3K15 context, there wasn't any other clinical risk that seemed to pop up even beyond 10 to the -4, so quite a liberal p-value threshold. There was, however, and this is where it gets a little bit interesting, earlier studies in mouse models that show that MAP3K15 knockouts in mice appear to have potential increased hypertension. And so we really wanted to probe into this because obviously if you increase risk of hypertension, then that's not something you necessarily want to be pursuing as an anti-diabetic. And so we looked at the human genetics data and we looked at males and females separately, and here conveniently, because it's X chromosome, homozygous males and heterozygous females. There are about 4 homozygous females, so we haven't included those. But what we see is that looking across the various phenotypes, so systolic blood pressure, diastolic blood pressure, and clinical diagnosis of hypertension, there was no evidence of an increased risk of hypertension. And this is all, of course, corrected for blood pressure medication. And so this then is interesting from a human genetics perspective. We don't see this safety liability in the in vivo human setting that we saw in the preclinical mouse, but it does bring other challenges about, this suggests that maybe in the context of MAP3K15 diabetes, the mouse model, which is very convenient and accessible, is not a directly translatable model for preclinical studies. And so that brings itself other challenges. And I think this is where we start to get into that next phase. So, the human genetics is increasingly, and especially with the larger collections of sequence data, allowing us to understand causal genetic discoveries a lot more confidently, a lot more regularly. Where we have the bottleneck shifting towards now is understanding what are the molecular mechanisms of those genetic perturbations in the human system. And this is where we have found, and others have found, adding other layers of omics data on the same individuals where you have genetics and clinical outcomes has been quite powerful. It helps us accelerate our understanding of mechanistic impact of those perturbations. In some contexts, helps accelerate clinical translation by identifying potential biomarkers of these molecular subtypes of disease. And it does deliver a competitive advantage because having these data sets available prospectively, proactively, allows us to increasingly answer some questions that previously took months to years to develop research plans and projects. To answer these questions, we're able to do them increasingly at real time, which accelerates the whole process. And I just want to highlight one example that we have found quite powerful, and I think most of you are familiar with this. So this is, again, another one of those pharma consortia, 12 industry partners come together with UK Biobank and Olink to generate Olink 1500 Explore data on about 60,000 UK Biobank participants. And this data set, it's plasma proteomics, allows us to answer various questions, proteogenomics, what is the impact of genetic perturbations we see on the plasma proteome, but also potentially in the context of blood-based diseases more so, and maybe some immune and inflammatory diseases, biomarker components as well. Maybe less so for diseases that manifest in other solid organs. And so I should just briefly mention, so this study and the consortium effort is well summarized in a preprint by Benjamin Sung, Chris Whelan from Biogen, which is kind of a consortium-based preprint on the array-based data. And we've focused a little bit more on the sequence-based data and looking at the kind of area of the site frequency spectrum that is less accessible through even the imputed array. And we're finding similar stories there. And what was quite exciting was that if you look at the XWAS variant signals, 97% of the protein truncating variants have a cis effect of reduction. There's 3% that seem to, for various, one could discuss reasons, increase levels, but the overwhelming majority decrease, and that gives us great confidence in the assay. In trans, however, that seems to be a bit more equal. So loss of function protein truncating variants of GeneX seem to have a more equal distribution of increase or decrease effects of other proteins in trans. And again, this data from the exome sequencing component, just to highlight, is linked with our preprint as a portal. So you can type in your favorite gene or protein and understand, based on the rare variant or the protein coding variants, what are the impacts of perturbing one gene on the roughly 1,500 assayed proteins from the Olink Explore. And finally, just to kind of take it full loop, so these data that we're generating, experimental readouts, if you like, can then feed back into discovery. And so one paradigm that we thought would be cool to adopt is taking those individual variants that achieve significance, missense variants that achieve significance in the same direction that we see as PTVs in those genes, and adding them to our PTV collapsing model. And so effectively what we're doing is identifying all missense variants which have a cis-pQTL reduced level reduction, and including that to our PTV models and seeing what is the effect when we look at that in terms of the gene-disease relationships. And one very profound one, which was highlighted, is PCSK9. So effectively, PTVs alone, it was already unequivocal. It was 10 to negative, 15, 16. But adding these hypermorphic loss of function missense variants based on the Olink experimental readouts boosted that signal to 10 to negative 110. So really empowering, and that is a very outlier one and a very good exemplar. But there are others where there were more modest effects, but still sufficient enough to tip signals into the discovery range. And again, I think this is going to be something that's going to be increasingly exciting as we generate, either through proteomics or other experimental assays, insights into what are the functional, saturation gene editing is one example, what are the functional effects of missense variants, and then feeding that back into some of these gene-based analysis paradigms. And I'll stop there, but of course thank again all the biobanks and participants and investigators that have made these resources available. In my presentation, I've introduced UK Biobank, Mexico City, and FinGen, my past and present colleagues at AstraZeneca, and again all the industry partners and peers that have joined this, have the same vision as us in terms of generating these data for the benefit of global R&D.
Thank you so much. Join me in thanking Slavé. Thank you. That was a fantastic talk. We do have time for a couple of questions, both in the room and online. So just a reminder that if you are joining us remotely via the live stream, go to slido.com, enter 5066. That's the winning number, and you'll be able to submit your questions for us there. I'll take a question from the room if there's any. We do have about 2 minutes or so.
If I may.
Yes, please.
So your work is fantastic, and you're really sort of plowing forward in genetic discovery, and you're giving us compelling examples of success. From your viewpoint, what would you see as the singularly largest sort of bottleneck for the type of work you're trying to do in the field today?
I tried to touch on that. It's definitely understanding more about what do these causal discoveries, we're at that point now where we're finding more and more causal discoveries. What do they have? What effects do they have on the cell, the tissue, the organs, the disease? And if you look at, for example, the FEWAS browsers that have been made available, there's so many discoveries there. It's impossible for any one organization to really, mine into all those to the right levels. So making them available and then benefiting from the kind of global communities, further deep dives into the biology, etc., will only benefit all of us.
Thank you.
Okay, thank you very much, Slavé.
Excellent.
If there are no more questions, I'd like to introduce our next speaker.
That's great.
Professor Andrés Moreno Estrada is a medical doctor by training, and he pursued a PhD in evolutionary genetics in Barcelona before doing his postdoc at Cornell University and Stanford. In 2015, Professor Moreno was appointed as principal investigator at the National Laboratory for Genomics for Biodiversity in Mexico, where his work integrates genomics, evolution, and precision medicine in understudied populations, particularly from the Americas and the Pacific. He's the co-founder of the Latin American Alliance for Genomic Diversity, and he is a member of the executive committee of ICDA.
Great, thank you, Claire, for the introduction, and thank you everyone for being here and for the opportunity to present on an exciting topic. It's really exciting to hear so many promising advances as just Ben laid out, and we just heard also from successful examples of what can be done in medical genomics, but also, it's a privilege to be part of ICDA. Together with many other members from other regions of the world. But I think it has to be highlighted that we are getting together precisely because there are big challenges that need to be tackled. And I think one of the biggest challenges is actually the representation of populations across the globe, because these advances that we're just hearing about, that we hope to take part of globally, are not equally distributed across different regions of the world. And I think, from my personal perspective, I can even, talk from personal experience that, I was born actually in an indigenous community in southern Mexico. My father, Arturo Moreno, he worked for many years as an anthropologist in many regions of Latin America. So I grew up really exposed to this reality of, health disparities, inequities on a daily basis. And that is reflected at all different levels. So these things have been appreciated by many disciplines, including anthropology. But to me personally, it's really contrasting to see that when it comes to genomics and this big promise that genomic medicine is, really genomics is not doing a really good job at it. I mean, we all know that most of the participants that are currently represented in genomic databases are mostly from European origin, and this really skews our understanding of human diversity, not only for Latin America, but many other regions of the world that have not been represented and keep being left out. This is not only like a snapshot in time, we can even follow this in real time with this GWAS diversity monitor, and we can actually see that this problem is getting worse. So if we compare, for example, from 2008 and last year, what is the diversity of populations or individuals being represented in GWAS, for example, if we look at the map, well, things seems to be getting better because there's more representation of regions, and that's good. It's actually, in absolute terms, a step forward of being represented. There are more countries and more populations being there, but proportionally, it's actually shrinking. If we look at the left, at the proportions of which are the number of participants of different ancestors, we see that actually all other non-European groups are actually getting a small representations as we move forward. And this is because even the larger initiatives are getting so big in terms of scale that even though there's some minimal representation from other groups, it proportionally is not big enough to really overcome this bias problem. So this is reflected also in larger diversity efforts, such as the 1,000 Genomes Project, which it's a great resource to have a reference panel across diversity of the world, but also has spots that lack representation, and I will point again to Latin America and the Pacific here, where what we learned from this study is that actually outside of Africa, Latin American populations, which are highlighted here in these red circles, are actually the group of the world that contributes the most in terms of number of new variants being represented by each new sequenced genome. And this is actually actually the group that shows the highest variability in terms of how much variation is contributed when we sequence individuals. And this is due to several factors. A major one is actually the degree of admixture that we know that characterizes the region because of the confluence of these different ancestry, that some of the segments of the genome will contribute as many variants as an African genome or indigenous American ancestry and some others of European descent. So this mixture really makes that Latin American populations are really a big mosaic of diversity that actually we can have a look at how it looks when we really look at genetic data. And this is an example from the PAGE consortium that characterized more than 100,000 individuals with reference panels highlighted here in color. And in gray is all the individuals from the PAGE cohort. And then if we ask, well, all the individuals that self-identify as Hispanic or Latino in this plot, where would they be, being clustering with? Well, it turns out that when we highlight that with H, as denoting Hispanic, they are actually covering every single corner of PCA space. Again, highlighting the fact that Hispanic or Latin American populations cannot be represented by a single label or do not represent a homogeneous group. It's rather a really diverse group that need to be studied, region by region in each of these components. This is important not only for anthropological reasons, it's also relevant for gene mapping when it comes to disease association. And again, this same consortium, the PAGE paper, discovered many loci that are actually new, highlighted here in yellow, that you can see across different traits how these traits or these loci have been discovered thanks to the inclusion of these multi-diverse cohorts. Actually, what they did is to map which of these new loci were associated with particular component or principal components in PCA and which population is driving each of these signals, as you can see in these two plots. And at the bottom, we can see that, for example, some of the novel loci are actually driven by specific populations such as Native Hawaiians of Polynesian ancestry or the indigenous Americans like PC4 and PC6 that otherwise wouldn't have been discovered if they were not included in the multi-ethnic GWAS. So this is really important, again, because we know from other disciplines, as I pointed out in the beginning, from anthropology, that and linguistics, there's a really high diversity and ethnically diverse population across Latin America that really deserves fine scale attention. And this is really what has been motivated our work for the past 10 years or so, and we have been conducting regional diversity projects across Mexico, the Caribbean, South America, and even further into the Pacific. So I will just mention very briefly the example that motivated really many of this work, which is the Native Mexican Diversity Project that started when I was at Stanford, but together with other collaborators across Mexico, geneticists and anthropologists, were able to study nearly 20 different indigenous groups and genotyping with high-density SNP arrays. And one of the first things that we learned about the diversity in Mexico is that it is really highly substructured, and there's a lot of isolation when we look at the genetic composition of each of the indigenous groups. So here, each dot denotes one individual, and the lines connect each pair of individuals based on genetic similarities by IBD segments. So what we can see is that there's really a general pattern of very few connections between groups and high levels of isolation and little gene flow between groups, which would anticipate a highly differentiated population, which we can actually measure by FST. Here we have a pairwise table measuring how much differentiated these populations are. And you can see blue colors denote low FST values, as expected, because we are comparing populations from not only the same continent, but the same country. And it's surprising what we found is that in some cases, when we compare, for example, populations in the north versus the south, such as the Seri population, which is an indigenous group in the north, the northern state of the Sonora Desert, versus the Lacandon jungle in the southeast, which is this red dot in the corner, we see that the FST value there is actually close to 14%, which really is higher than what we'd expect even within the same continent. For comparison, I put at the top was the value that we get when we compare Europeans versus East Asians, which is 11. And this is even higher than that comparison. It's speaking again to these high levels of isolation, differentiation, and therefore genetic variation that may be private to these populations. So the big question was at the moment, okay, then what is the contribution of these highly substructured indigenous ancestry roots into the general population of the country? And this was done in collaboration with the National Institute of Genomic Medicine back then, which were collecting urban samples from many of the states colored here, highlighted here in color. And then we put together both data sets to see what's the correlation between these genetic profiles. So the picture that emerges is actually that there's a big correlation between the ancestry components from the indigenous Americans in blue, and you can see when we break down the model to allow a higher number of groups, actually they contribute proportional to the different states on the right. They are like from north to south organized, and we see that regional components such as the light blue, which is mostly represented by northern indigenous groups, is mostly represented by northern states in Mexico, and so on. The Mayan region, for example, in the Yucatan Peninsula, mostly has this orange color component that is also reflected from Mayan indigenous groups. So this again speaks to the correlation between these two. And we wanted really to develop a method that allows us to numerically explore it and really put a measure on those segments that are of a specific ancestry. And this is the so-called ancestry-specific pipeline of analysis. Initially, this was done for PCA, but it can be implemented for many other approaches that we call ancestry-specific. So the basic idea is that we are able to assign locally, which is the ancestry that is assigned to each chromosomal segment, and then we mask out all the other ancestry that we're not interested in analyzing, and then combine it with further reference panels to explore more what are the origins or the affinities in those particular segments. So what we learned from this first study is that the Native American fraction of the genome of Mexicans is actually correlated with the geographic location of these cosmopolitan samples, and that they actually reflect a geographic map of Mexico when we look at where they have been sampled and their genetic affinities in PCA space. So this is not only interesting for the purpose of knowing the structure of the population, but also that it correlates with biomedical traits, as we demonstrated in the study in 2014, correlated with lung function measures. So this really motivated the fact that we needed a more representative set of samples nationwide and across many more biomedical traits if this holds. And that's what really motivated the fact that we needed to launch a bigger initiative, and this is the so-called Mexican Biobank project. And this is now a more representative collection of samples in collaboration with the National Institute of Public Health in Mexico that conducts national health surveys every given year in Mexico. And for the edition of 2000, they actually collected more than 40,000 DNA samples together with associated biomedical traits. In a first round of funding, we were able to genotype more than 6,000 samples so far, and this is in collaboration also with the University of Oxford and several other institutes across Mexico. So the next slide just shows you the sampling location of individuals, and again, just the fact that the coordinates reproduce a geographic map of Mexico tells us how nationwide representative this is, both in rural and urban locations. And like I said, the data set has also associated traits, including measures of each individual, and then also biochemical measures in blood, and also we have measures of immune response to different pathogens. So we're conducting a different set of analyses. This was mostly led by one of my former postdocs, Michelle Sohail, in Mexico. And one of the things we have been exploring is what is the correlation between ancestry components, like I said, in this mixture of the biobank and each of the functional categories of different mutation burdens. So for example, intergenic, coding synonymous, or deleterious. We see that there's a clear correlation in all three categories according to the segments being called European, Indigenous American, or African. That tells us also how the demographic events in each of these components is affecting the load of mutations in all three categories. And to give a more clear example of how this can be correlated with biomedical traits, we model simultaneously different genetic and non-genetic variables, including environmental, age, sex, and so on, in different medical traits. Importantly, besides the obvious ones that we found, like such as height, that is clearly correlated with ancestry across Mexico, and this is mostly reflecting the indigenous American ancestry. Also others like triglycerides or BMI are correlated with other genomic factors such as ROH, or the fraction of the genome that is in homozygous state, which also tells us that it's important these bottlenecking evolutionary forces that have been affecting Mexican populations. We are also conducting a series of GWAS, and we are able to replicate a number of loci that have been reported to be associated with lipid values, especially cholesterol. But what is important to highlight here is that for more complex traits, we were not able to find any significant hit, mostly because of the lack of sufficient power. It's still a fairly low subset of the biobank, like I said, 40,000, and we are only genotyping so far more than 6,000. Another motivation for this resource is actually to create a common set of controls, similar to what the Wellcome Trust case control study did several years ago, that we can actually provide a resource to the community so that they don't have to spend again in genotyping controls for the population and only focus the resources in those cases that they want to study, and that they can use the Mexico Biobank as a public available control set. Importantly, I want to highlight that this is also mostly done, all of the stages of the project have been done locally, and from the biobanking, the preservation and the maintenance of the samples, the local generation of data, the genotyping has been done at the local laboratory in where I work, and also data analysis has been conducted locally, and then of course data analysis in collaboration with all the members of the consortium. So this really motivated the fact that we need to look beyond what the diversity of these regions is. Because if we are demonstrating that there's such a high level of diversity within a single country, we expect to see similar patterns in different regions, and that's the case. If we look at other populations in Latin America, like here, I'm showing the distribution of Native American segments of other South American countries, highlighted here in color, and look at the distribution of their genetic profiles do not overlap with what we would expect in central Mexico, highlighted in green, what we call their Central Native American. See that the space is really not even overlapping with the profiles of other countries in South America that also fall more or less closer to their ancestry references. Like, for example, Andeans are the closest reference for Peruvians, and Amazonians are the closest reference for Colombians, and so on. So this really speaks to the need of addressing these different populations, kind of one by one. I would mention just examples in one of the South American projects we recently published this year, which was GWAS on preclamptic women from the highlands of Peru in the city of Puno, which is more than 4,000 meters above sea level. And we collected nearly 1,000 cases of hypertension during pregnancy. And what we found is actually that there's one region in chromosome 13 where several clotting factors are associated with this condition, detected mostly through a TDT test, which still was quite low for genome-wide significance in a standard approach, but again, speaking to the need of these specific discoveries that can be done with populations that have not been studied before. And we suspect that there's also some adaptive mechanism going there because of this higher incidence of preeclampsia at high altitudes. Another example that I should mention is also moving into other collaborations in South America is, thanks to Ricardo Verdugo, also here from Chile, is this partnership with the Chile Genómico Project, which has a similar objective as the Mexico Biobank, a nationwide initiative to characterize the Chilean population. And this really led us also to include other populations that are geographically far away from Latin America, but that are also part of the country, such as the Easter Island, as you know, has Polynesian roots, which actually led us to study other parts of the Pacific, which is now growing into this Oceanian Genome Variation Project, which is actually one of the biggest collection of DNA samples from all across Oceania, near and remote Oceania. And from this study, actually, we have been learning several things already about the diversity of this other region that also is heavily underrepresented. Mainly two things we have been reporting so far. On the left, we have the first paper that allows us to recover the paths and the split patterns of the peopling of all these highlands across the Pacific. And on the right, we reported the fact that we found also genetic components of integration and gene flow between indigenous American ancestry into Polynesians before European presence there. So these two papers have been published in Nature in 2021 and 2020. And this is interesting for anthropological purposes, and this is really exciting. But also the fact that we have this collection of DNAs from samples that have not been studied has led also to medical discoveries. For example, in collaboration with Luis Quintana Murci from Paris and John Laurent Casanova in Rockefeller, we were able to genotype a variant that is a loss of function in the receptor of the interferon that is private to Polynesian individuals. This was discovered through cases that were affected by viral diseases, and actually when they are in homozygous state, we found that they are severely affected, and this is a variant that is exclusively present in Western Polynesian. Again, wouldn't have been discovered if we were not able to characterize what is the distribution of this variant across different populations, and we can tell that this is important now for diagnosis and screening in terms of epidemiology purposes. So This is really where it comes to the importance of responding to the recommendation of ICDA, as Cecilia pointed out in the beginning, where there's really an importance of increasing the diversity of existing biobanks. And this is how the Latin Genomes, or more formally named Latin American Alliance for Genomic Diversity, really comes into place, and you will hear from Ricardo in a minute what the specific proposal is. But in general, the idea is to contribute to all three goals that ICDA has been mapped, which is moving from maps to mechanisms to medicine. I'm just going to mention very briefly what are the steps that we're taking forward to contribute to all these three goals. In terms of maps, I already mentioned the existence of the Mexico Biobank Project, the Chile Genomical Resource. There are others initiatives like in Argentina, like the Polar, which are resources that are available to be already scaled up and sequenced to provide a better reference panel for many projects. And also, talking about this other layer of the single-cell resolution of gene expression, I would mention that we have a pilot project funded by the CZI initiative that is allowing us to contribute to the Human Cell Atlas project in several Latin American populations. I would mention that this has, at the beginning, the idea of collecting PBM cells, tissue samples from across different locations in Latin America, but also tissues of a new network that will be contributed, which is gallbladder, particularly because in Chile, there's a high correlation between Mapuche ancestry and the incidence of gallbladder cancer. I mean, briefly, I won't go into the details of the technical methodology, but basically, we will, follow a similar pipeline of biobanking these samples for single-cell sequencing or single nuclei for the frozen tissue, and then after applying methods, we will also develop a community portal for people to get trained and analyze the data. This is also to mention on one side on the left that really the project is just a pilot, has limited funding and limited number of samples that can be collected in this first phase. That will also have the potential to contribute to the other projects have been highlighted by Ben and others, like BodyMap or Disease Map. And this really is an opportunity for being scaled up. Once the network is in place and we have been able to get samples, this can be scaled up if future rounds of funding are available. Actually, I'm very proud to mention that just last week, we were able to standardize the protocol for isolating PBMCs on site. This is physically taking like a little molecular laboratory to rural areas. And actually, we implemented a method that allows us to isolate the cells from site, keep them frozen, and get them back to the lab for further analysis. And this is done, in even remote areas, we can transport them back to the lab. And we are ready to then, actually sequence them locally, because I also would mention that this equipment from the 10x Genomics that allows us to construct single-cell libraries has been just recently installed, and then the same thing will happen in the next few weeks in Brazil and Chile. So again, this is proving the capacity of doing things locally. And finally, the other major goal of Latin Genomes is to really aggregate biobanks that are either existing or we can create new ones. I won't go into the details as you will hear from Ricardo later on. But the idea here is to take advantage of these existing network collaborators that have already access to these medically relevant cohorts and aggregate them or escalate them in different disease focused. The overall idea is to contribute to this landscape of biobanks, not only in the fact of, like I was saying at the beginning, of being represented with absolute numbers, but in terms of scale and volume, we need to really take this to the next level to be able to make equally important discoveries in underrepresented populations as it has been occurring already in other parts of the world. And I would say that Latin America is ready to do this. I mean, we have the infrastructure. We have been demonstrating community engagement. We have been training people. So, I mean, really, it's just a matter of getting closer to that next step. And I would say, well, then, if I said that everything is ready, then what is missing? And I think we can agree with Eric Green, for example, that what is missing is diversity. And as I was said in the beginning, we need to really address this problem. Where we probably don't agree is in the priorities of this, because we just responded to the NIH RFI with a proposal to increase diversity from Latin American populations, which actually was rejected and saying that they cannot take this idea for the moment. Hopefully that will change in the near future. But then what I think is really missing is funding, like I said. It's not a lack of talent, it's not a lack of motivation, it's not a lack of local capacity that is really to be scaled up, but we need the proper funding and really put Latin America in the top of priorities of funders, and hopefully this meeting is what it can be about. Thank you very much.
Thank you, Andrés. That's a tour de force that you're sort of mapping out eloquently over 13 minutes, but as you said, it's 10 years of work to get to where you are today. So we have time for a couple of questions in the room. People are jet-lagged, maybe? And for those of you who are online, go to slido.com. 5066 is your number. Do we have any Slido questions? So I have a question. If you think about the infrastructure that you're setting up, similar to what I asked Slavé before, what do you, beyond funding, which is obvious and you've mapped out, what do you think is the singularly sort of most exciting part that you haven't done? Because you mentioned you have talent and you have infrastructure and so forth. What's the sort of key excitement for you that you would really want to get stuck in with if you had to choose one thing to take forward and sort of kickstart with?
Sure. Well, I'm sure you will hear more from Ricardo in a few minutes, but I think we really still don't understand what is the extent of novelty that it's out there to be discovered, right? I mean, we know that there's a lot of diversity, these highly substructured populations that I'm sure they're really holding some key discovery that we're still not able to see because there's just not enough data from many of them. At the same time, there has been a lot of efforts, mostly focused where there's more feasibility to be being conducted. And then it was already mentioned that the Mexico City study is one of the biggest resources in terms of volume. That it's really great to be to have that resource. But at the same time, as I demonstrated, we have way more diversity outside of one city. So just in one country like Mexico, we have a lot of diversity that has not been represented at the scale that we need to discover rare variants. And we don't know what is the implication of that in the whole chain of these mechanisms and potential medical pathways that can be explored. So I would prefer, to have a highly diverse represented biobanks at the scale that we needed to explore all these options that can be taken all the way to mechanisms and medicine, not just mapping.
And we know from the field that special population have been incredibly useful, looking at Iceland and Finland, for instance, so that we know that there is a lot to discover in these settings as well.
That's right. Population actually definitely have that opportunity. Nicole? Yeah.
Nicole.
So I mean, just mind-blowing. I guess my question is, there's a lot of genetic...
I'm Nicole Soranzo from Human Technopole Sanger Institute. Yeah, it's just mind-blowing work. Really, congratulations to you and your colleagues. I'm just thinking, there's incredible genetic diversity clearly and structure, but also amazing environmental diversity, right? Here, these are huge regions, north to south in Chile, completely different environments. So to what extent are you trying to capture some of these environments, and or to what extent do you think this is going to be important to do? So that's the first question. And the second question is more on the capacity building, needs, again, building capacity, bringing capacity locally, and empowering local communities to work in genomics. So where do you think are going to be the most effective interventions at this stage?
Right. No, a couple of great questions, Nicole. Thank you. I would say that, definitely this level of diversity really tells us that we need to go at the higher possible scale, right? I think in terms of potential, having the highest impact would be those places that have already some intermediate level of capacity, so that is not that difficult to be starting from scratch, but that need to be scaled up to the next level. For example, areas where recruitment has been proven to be possible, and then just being added to this prospective component that needs to be included. Because so far we have efforts that have been just like efforts that have been a snapshot in time, but not really with this component of follow-up that is really what I think is missing. On the second question, and the first one, if you can remind me the first one, sorry.
It was about environmental diversity.
Yes, which is definitely why we have to stress the fact that it is not enough to collect these ancestors that may be represented, for example, in other urban areas like in the United States or like in the UK with similar migrant communities, right? I think environmental really is a key component that would allow us to understand what is the genetic factors affecting the outcome of the disease. And this is something that, again, taking these examples of places that can be doing this prospective recruitment, can be really incorporated. We do local recruitments across diverse regions in Latin America. And it is the only way to really capture those environmental factors. This is why also why I mentioned that in the Mexico Biobank, for example, there's biomedical traits associated with the genetic data, but we just are not there at the scale that we need to grasp really all the genetic basis of those associations. But really is the way to go to keep recruiting locally with all the possible variables we can collect. That is only possible if we do it locally. And with other many reasons, which is also the capacity and the fact of controlling the access of those data by people that is leading the study locally, besides the contribution that can be done globally.
Any other questions? We have one question over there. The microphone will migrate over to you, sir. And also go on slido.com, 5066.
Hi, I'm Jonathan Pollack from the National Institute on Drug Abuse. So a great presentation. So a follow-up is, a general question for biobanks is really how do we capture environmental variables that are lacking in all of the biobanks?
Well, definitely, I would say that deep phenotyping is the key, right? And definitely in many of these efforts, sometimes even most expensive part of what needs to be done. I think that many have proven that sequencing or genotyping is no longer the bottleneck. I think it's this deep phenotyping that has to occur locally with, like I said, prospective capacity of following people. And that also varies a lot, and you will hear from Ricardo different scenarios where we have been exploring different countries, which is the capacity they have. In Mexico would be maybe in one central hospital that have access to calling back individuals to measure locally different exposures. Costa Rica, for example, has a great example of really having this national system of collecting all the biomedical data in a centralized database. Chile will have a different situation, but I think deep phenotyping is the key for that.
I think also maybe interesting when it comes to the environment is that it's a very loose term that is very generally used, and what the environment constitutes will be very different for different constituents. It can be anything from smoking to the type of medication or drugs you're taking, to lifestyle choices, to the environment around us in terms of temperature and exhaust fume exposure, to radiation from building materials. So I think that the field really needs some sort of disciplined approach into thinking what the environment really constitutes and how we best measure it.
The National Institute of Environmental Health Sciences has a project called the Exposome, so that might provide a roadmap for how biobanks might incorporate some of that information.
So maybe Ricardo could sort of liaise and have a chat.
For the panel, right?
Yeah.
I was thinking that we can discuss that on the panel.
Yep.
Yeah.
Perfect. So I'm thinking especially our friends from far, far away are going to need some caffeine to boost up the jet lag and to stay awake and perky. We're going to have a 15-minute break now before we resume the session. So just take a moment to thank the speakers and thank you for your questions. Keep these on. Welcome back. And we're going to continue the GeneticMap session now. I hope you all feel refreshed and that you submit all your exciting questions on Slido with the code 5066. So the next talk is going to describe the plans in more detail on the genetic pitch from the GeneticMap, Latin American program to accelerate medical genomics in admixed population. It's going to be given by our own Dr. Ricardo Verdugo. He obtained his degree in veterinary medicine from the University of Chile, subsequently a PhD in genetics from UC Davis, before completing a postdoc in computational biology at the Jackson Laboratory and INSERM in France. In 2012, he was appointed as an assistant professor in the human genetics program of the University of Chile and became a group leader at the Institute of Interdisciplinary Research at the University of Tucker. Dr. Verdugo is the director of the Chile Genomico project and is a co-lead of the Latin Genomes Consortium within the ICDA Genetics Map umbrella, and also sits on our organizing committee. So without further ado, welcome.
Hello. Thank you, Cecilia, for such a kind introduction. And I'm really privileged to be here in front of you. But I'm just a messenger for a large group of people down in Latin America that have been thinking about how we better represent genetic diversity in that continent in global efforts. So what I'm going to present to you is a product of over 2 years of discussions with other colleagues in Latin America, and we came with this name of AdMixMap for a pilot project that we think is really necessary in order to make a change in this pervasive problem that we have heard already by Andrés of the underrepresentation of Latin American diversity in genomic studies. We know that increasing representation was deemed important by ICDA in its white paper, in mentioning particularly the Americas. And so we wanted to propose an approach for this. And we take as a great example H3 Africa, which I don't need to introduce to you how successful this project has been, other than saying that if it made it to be the front page of Nature, it's because it has proven on the promise of increasing the number of discoveries by studying other populations than just the few that have been studied over and over. But the key to its success, we believe, has been not only large funding, but also the infrastructure that has allowed such discoveries, because they have not cared only about recruiting individuals, collecting samples, and genotyping, but also about other implications of this type of research in that continent that will give it sustainability by promoting local capacity. They created the H3 Bioinformatics Network along with H3 Africa, so that now they are empowered to study their own genomes. And I think that has been critical because it allows for a long-term planning for making new discoveries in such a large and diverse continent. So we asked ourselves, after our previous meetings in Copenhagen, Washington, why can we do this in Latin America? What are the barriers, the gaps for us having an H3 Africa-like project in our continent? And so the first thing that we did was starting asking our colleagues across different countries in Latin America, what is the status of things? What are the projects that are ongoing? What has been done and what can be done? And from that process, we started a new network that is called the Latin American Alliance for Genomic Diversity, which is very much associated, independent and has a common vision with ICDA. And from this process, we realized that there were a number of national initiatives across Latin America that with sometimes very little or no funding, were able to survey genetic diversity in a scale that has not been recognized internationally. And here I'm just showing some of those initiatives, like the ones that have already been described, the Mexican Biobank or Chile Genomico, but there are large initiatives in Brazil as well that are way advanced compared to other countries, and new ones like Poblar in Argentina that are starting to survey genetic diversity there. So we started with a survey asking the current status of previous work and ideas for new projects that could make a change in Latin America for leveling up, for increasing the level of studies there, and also estimated cost of such projects. And from this survey, on our colleagues, we came up with summaries about gaps and opportunities to address in our continent. And without going into the details, we can see that the realities are diverse across countries in Latin America. Some countries like Brazil have a high level of access to funding and infrastructure and are more advanced. Others lack those resources, but have a high level of local trained professionals, either in healthcare or access to health records and so on. So the actual solution will have to be diverse depending on local realities. I'll just skip this one. So in summary, we detected at that survey 38 different projects that total 100,000 samples that have been already collected, 18,000 arrays that all have already been generated, over 4,000 whole exome sequences, and over 1,400 whole genome sequences. Although most of those whole genome sequences at that time came from one country, Brazil. So there is potential. There are already projects that can support a large-scale genomics project in the region. But also, there is established infrastructure that has allowed such discoveries. There is expertise in bioinformatics, and there is local capacity for generating genomic data across different countries, but also a lot of experience in community engagement, which we think is critical in order to have sustainable research in the region. And also previous experiences in training, in local languages, in genomics and bioinformatics. This slide has been shown already, but I wanted to bring this up again because they are part of this, the framework in which we can work on and scale up. So the Mexican Biobank, as was mentioned already, has genotyped over 6,000 individuals, but could be scaled if funding was available to 40,000, capturing genetic diversity across Mexico that we have seen already that is quite diverse. But also in Chile, the Chile Genomico project has already shown that there is high diversity between the north and the south of the country. We already have 500 individuals genotyped, but could scale up to 3,000, and we are currently recruiting individuals in Patagonia, which wasn't included in the first phase of the project. And Poblar is a new initiative in Argentina that is starting a similar study there. Brazil, as I mentioned, has the largest number of resources already, with a large nationwide project called Genomas Brazil that is capturing genomes from different cohorts and has a target of sequencing over 100,000 individuals. And in conclusion, the reality in Latin America is diverse. The solutions will have to be tailored to the realities of each country. But there is a baseline from which we can start. There is already knowledge about recruitment and engagement, and there are some biobanks already established with high standards. I'm going to talk a little bit more about that. And but there is basically a lack of long-term funding that could allow large-scale projects like H3 Africa. So what we propose is called AdMixMap, which plans to generate reference panels, a network of cohorts, SQTL mapping panels, and a network of biobanks. But this is sustained on 4 pillars, which are community engagement, data sharing, local capacity, and sustainability of these research programs. So I'm going to talk a little bit about each of these planned outputs. So for reference panels, we have already heard about our ongoing project funded by Chan Zuckerberg Initiative to contribute to the Human Cell Atlas with Latin American diversity. But as you can see here, the sample sizes are quite small, but we are already oversampling so that the samples will be collected. And if funding was available, we could increase those numbers and become a significant contributor to BodyMap. There is also still a need for a good reference panel for imputation that could be used for GWASs, right? And as Andrés has shown already, you cannot just put all Hispanics and all Latinos in one bag and expect that they will behave as one homogeneous population, because they are not. And they are the process of an admixture of multiple populations with large genetic diversity, comparable to Europeans versus Asians. So we need a panel that accounts for that diversity, and our alliance, the Latin Genomes Alliance, already has compiled a large set of samples from all across the continent that are that could be sequenced for creating such a panel. In addition, we have already started networks of cohorts. In particular, for COVID-19, we have pulled together resources created by different sources of funding with the common objective of finding genetic variation for either susceptibility or severity of COVID in Latin America associated with its particular ancestry. And this has already given us some results where we have already replicated, some major peaks of, in this case, severity of COVID in chromosome 3, but we have also identified novel peaks that are barely below genome-wide significance. That is, if we apply the same threshold as Europeans, although that may not be the best, because we don't know how many independent hypotheses there are in these types of genomes. But we are already identifying new loci, and if we could scale up these efforts, then for sure we could provide new discoveries. And I just wanted to mention an example of how we are doing this already in Chile. Until a couple of years ago, there was only one biobank in Santiago, the capital, and we are now creating a network of biobanks that go from Arica in the extreme north to Magallanes in the extreme south, all using exactly the same protocols, standardized and harmonized data, and using the same recruitment protocol with SOPs and pre-label enrollment kits, all the staff being trained across the different sites so that everything is done harmoniously. And we plan to expand this to the whole to the continent by creating a network of biobanks totaling 1 million individuals. That will include enhancement of existing biobanks, the creation of new cohorts that are population-based and not oriented, not biased to any particular disease, and then using those two types of resources for multi-omics profiling, including genomics, transcriptomics, and proteomics, in order to create SQTL mapping panels that are open to the larger community. In terms of phenotyping, we have selected those phenotypes that are most common in the region and that are most relevant, but also that are relevant to the larger community. Of course, this will be done harmoniously using tools like RedCap, but also will be profiling molecularly samples that will be collected here for common diseases like cardiovascular disease, dyslipidemias, diabetes, and some mental disorders, along with some autoimmune diseases that are prevalent in the region, like rheumatoid arthritis and IBD. In terms of biobanks, we propose to create a network of biobanks, and we have identified a number of gaps in the region to do this. I'm not going to go into the details because this is a busy slide, but we have identified also opportunities for performing this type of studies. We recognize that the concept of biobank even is not that well established and accepted among the healthcare professionals and the governments in Latin America, and that has been a problem. So, and we are working in the process of also educating professionals and public health officials in the importance of having available biobanks. And we think that so far we have been successful. We have been participating, at least in Chile, in the last year, in a long process of creating a regulation that is now going to be seen by the Congress to allow for the regulation of all biobanks across the country. And in terms of sustainability, we are also believers of the value stakeholder model, where we do not depend only on government money, but also in a relationship with the private sector that would allow for continuous funding of this type of initiative, which is a must. But in order for this to have value, it has to be done properly. And we believe that that accommodating standards is very important for having good quality data and samples, and that is the way that we are doing it in Chile right now, and that we are planning to extrapolate to other countries. This has given results. We can measure the quality, even the time of processing of all the samples across the different sites, so that it maximizes the use of the material. In terms of the methods for our approach, we plan to have a board of scientific advisors that will help to start this initiative and keep it in course, but also we recognize the experience that we already have about doing genomic research, but being conscious of local realities. We have existing cohorts that we can expand, and that we can use to create genomic and multiomic resources, along with some methods, and we will include national initiatives that are already linked to known linguistic information, ethnic, geographic information, and so on. So I'm going to skip this. In summary, what we are proposing is creating a network of cohorts and biobanks that, network of biobanks, mainly in urban areas that would allow long-term follow-up, and this along with good quality stored samples and genome data for admixed genomes, and instead of sampling isolated small populations that will mix genomic with local environmental variables, the power of mixing the genomes and the environments in one area would allow us to now not only paint genomes, but also paint pathways in terms of ancestries. And these new combinations of different ancestries and pathways would allow us to perturb the system so that we can identify new states of the system that cannot be identified in each of these ancestries isolated. So this will allow us to contribute to the body map, disease map, and why not the blood cell map or the cell mapping efforts for new discoveries. But very importantly, those discoveries cannot end in publications. They have to be returned to communities so that the communities can stay engaged in these studies, and we can ask new questions in the future. And I think this is what distinguishes our proposal from others. And with that, I'll finish. And I'll take any questions. Thank you.
Thank you. So I'm going to ask if we have any questions in the room. Otherwise, I'm very excited that we have 3 questions on Slido. So I will start with Daniel Corral. I don't know where you are, Daniel, if you're online or here, but he's asking if you have any plans to link genetic data to electronic health records.
Okay, so electronic health records are not that common in Latin American countries, and as opposed to, for example, the UK, there is not a common system in most of these countries where all of those records can be found. So that's a challenge. The situation is better in Brazil. There is already efforts for doing that in Brazil. And in our case in Chile, we do it, but in a very artisanal way by collecting information by hand from the records and then moving that information into a REDCap form. It's a CRF. So that's a bigger challenge, and I think that we cannot base the whole process on health records yet.
Great, thank you. So next person is asking, great talk. Can you discuss what phenotypes you have available and what phenotypes you can collect? What are the barriers to collecting phenotypes for you in Latin genomes?
So the phenotypes being collected so far depends on the cohorts. In the case of the Chilean cohort, which was based in COVID, of course we have everything related to COVID, but also socioeconomic information, demographic information. But we also ask for all sort of chronic diseases that were present before the onset of COVID, and now after 1 year we are asking them whether they have new diagnosis of chronic diseases, and that includes cardiovascular disease, metabolic diseases, cancer, mental disorders, and so on. And the advantage of having a longitudinal study like this is that we can then go back and ask again for more phenotypes. So I don't think that there is a limit in terms of what phenotypes we can collect. Of course, environmental information is much harder to collect. We do collect some of that, but we think that by using cell phones, we could do a much better job at that. And actually, we wrote a project to create an app, because everyone uses a cell phone in Latin America, so it's a great tool to collect physical activity information and some diet information, and that's something that we have in view. Yeah. I don't know. In Mexico, probably Andrés can tell us more. But other than COVID, diabetes is a large problem there that they are addressing in the Mexican study, and in Costa Rica, they also have high interest in viral diseases and some types of cancer and mental disorders, schizophrenia. So we are interested in very much all of those common diseases. Yeah.
It's been quite inspiring also to see, the power of questionnaire data, for instance, in 23andMe, that not always, but quite often the questionnaire-based information delivers genetic associations that have a very high genetic correlation with that of a much more bespoke phenotype.
Yeah. But the only way for that to be successful is that people answer those questionnaires. And they will do it if they feel that they are part of the study. Yeah. And that's what we have learned. In our COVID study, we had a success of 75% after 1 year of contacting everyone by phone and asking them to answer a new survey that takes about 10 to 15 minutes. So compared to other studies, we think that was a success, and we have a program where every year we return them results. We give them an ancestry report so that they keep engaged. For people that will go and take a blood test in the morning for diabetes, we give them a T-shirt with their genome printed with the personal, colored by ancestry. So we are thinking about ways so that people keep engaged in this type of studies.
Mm. Do you have the opportunity to recall people then? Do you have the rights to recall people?
Yes, that was done like that from the onset. The informed consent allows for recontacting, for biobanking, for data sharing, because it was started after we joined ICDA, so we couldn't do it otherwise, right? And so yeah, it all depends on whether people feel connected to the project, that they feel represented by it, and they want to participate. Of course, in the beginning, everyone wants to participate. We had so many people, thousands of people that wanted to participate. Now, cases of long COVID want to participate more than those that did not develop long COVID. So we have a bias there. But I think that COVID itself was a huge engine of motivation for people to participate in genomic studies, and also it helped to show the authorities of the value of having biobanks and having cohorts, because they could ask questions very rapidly. And I think that that message went through not only in Europe and the US, but also in Latin America.
Nico Vins also liked your talk, and they want to ask if you plan to include HLA imputation reference panels in your project.
A Chile imputation panel?
HLA.
Okay, HLA.
Sorry for my Swinglish.
No, sorry. Yes, HLA, it's actually is very important to us, particularly for COVID. So we are actually writing a grant for particularly sequencing HLA in the whole cohort, because we don't think that we can capture that region properly in the arrays, and actually we think that we have some false discoveries there because of some biases in the imputation process. So yes, we would love to, but we will need extra funding for that. We are not currently funding to do that.
Okay. Thank you so much, and thank you for all your questions.
Thank you.
Thank you.